Hello. So very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible, and also if you can see the video and the PPT clearly? Please let me know in the chat box if my voice is audible. Yeah, thank you. And I hope the PPT is also visible. So we will wait till more students join, an hour up to 6 p.m. Then we will start today's session. Okay, 6 p.m. I will wait for some time till more students join, and then I will start today's session. Let us wait for 5 more minutes and then we can start the session for today. Okay, meanwhile I can start with the introductions. So very good evening to the students. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry, and today it is the week 7 live session for this course. In today's week 7 live session, what we will do is sorry, we will continue the topics of week 8, because we are going ahead with each and every week with the courses and the contents. So last week in our week 6 live session we have done the course content of week 7. Okay, so today's class what we will do is we will continue the topics of week 8. And right now the week 8 is live, so these kind of topics and these kind of questions will help you to understand the course better, and also it will help you to solve the weekly assignments. So I will start in few minutes, so whoever have joined, please wait for some time. I will start soon, so when more students will join. Okay, I will wait for just a few more minutes and then I will start today's session. So, like every class, I point out few things before starting the session. So the things are like there will be this kind of problem solving session every week. Okay, and these sessions will be live on Google Meet platform. The code or the class code or the Google Meet link is the same for every class, and these sessions will be on every Tuesday from 6 p.m. to 8 p.m. So if any network disruption happens from my side, please wait for some time. Do not leave the session. Okay, I will try to join as soon as possible. And today itself I think there is some network issues from my side, so if there is any disruption happen, please do not leave the class. Wait for some time. I will join as soon as possible. Okay, so during the session the students can post their question, their doubts, their queries through the chat box. I am always available in the chat box. For the smooth streaming of the session, I have just kept your camera and microphones off, but I am always on in the your chat box. I am checking the chat box now every time. So if you have any doubt or any query, you can just let me know in the chat box. Okay, I will try to answer your query. And if still any doubt persists after class, you can drop an email to me. I always share my email ID and in the session. So if you have any doubt, any query, any problem, you can just drop an email to me. I will answer the query. And this presentation and class recording, it will be shared to the students soon. So you don't have to worry if there is any network issue from your side. So you can just rejoin at any time to the class. There is no problem. And the presentation and recording will be shared to the students soon. Okay. Today it is the week 7 live session, and right in this week 7 live session what I will do is I will continue the topics which are in our week 8 class content. Okay, so in that week 8 what the topics are there. So right now the week 8 is live, right? So in this week 8 the topics which are included, that is these. So the topics which are included in week 8 are the optimizing drug target interaction, which is divided into 4 part 1, 2, 3 and 4, and then we have a tutorial which is the tutorial number 12. So all these are included in the week 8, and right now this week 8 is live and you have to submit your assignments on this week 8 soon. So I think I will discuss now around 20 questions related to the week 8 topic or course content, and these questions, all questions will be MCQs. It will just like you have in your assignment portion. So I want all of you to participate, and whoever have joined the class, okay. So please participate and let me know what is the answer of that particular question, and even if your answer is wrong, it does not matter if because there is no marks in answering during the live session, but if you are answering, I will understand that you are following the class or not. So I hope my voice is audible. Before starting, I am just checking it out. I hope my voice is audible to you and you can see the PPT clearly. If not, please let me know in the chat box, okay. If you have any problem listening to my voice or looking at the PPT, please kindly let me know in the chat box. And if any network disruption happens, please do not leave the class. I will join as soon as possible, okay. So now let me start the session. So the first question of today's session is, which of the following statements best describes the structure activity relationship or SAR? Your options are: number A, the study of which functional groups are important to the chemical reactivity of the drug. Number B, the study of the physicochemical properties that are important to the absorption of the drug into the blood supply. Number C, the study of the structural features of a drug that are important to its biological activity. And number D, the study of the structural features of a drug that are important to its chemical stability. So which of the following statement best describes the structure activity relationship or SAR? Again I am repeating the options. Number A, the study of which functional groups are important to the chemical reactivity of a drug. Number B, the study of the physicochemical properties that are important to the absorption of a drug into the blood supply. Number C, the study of the structural features of a drug that are important to its biological activity. And number D, the study of the structural features of a drug that are important to its chemical stability. Which of the following statement A, B, C or D best describes the structure activity relationship or SAR? I will give you some time. Please read all the options very carefully, and in the chat box let me know A, B, C or D which of the following is the correct one. Which of the following statements best describe the SAR or structure activity relationship studies? I will give you some time, think, and then let me know in the chat box. Okay, let me check the chat box and then I will show you what is the correct answer and we will discuss about the SAR. Okay, so the answers are given number C and number D. Okay, so now let me check the chat box and then I will tell you what is the correct answer. So the answer are given like number C and D. So now let me show you what is the correct answer, then let us discuss what is the SAR. So the correct answer will be number C, the study of the structural features of a drug that are important to its biological activity. That particular study is known as the SAR or structure activity relationship study. So now let me show you about what is the SAR. So basically in case of SAR, what is SAR? SAR is like your structure determines the function. It is presented by the Crum-Brown and Fraser in 1865. Okay, let's say the structure of a compound determines the biological effects it has, and the determination of structure function relationship helps medicinal chemists to synthesize new drugs or pharmaceutical agent. So what happens during, basically let me tell you what is the structure activity relationship. So what happens, like we have a whenever we are doing a medicinal chemistry, okay, or whenever we are trying to find out a particular therapeutic agent, we synthesize a series of compound. Okay, so in that particular series of compound there should be a particular group which is fixed, which is known as the pharmacophore, and then we just change the functional group which are attached to that particular series. So let's say, for example, just look at this structure. So this fused ring system, which is A, B and C, so this is the central ring system, and there are various groups which are attached to this particular system, right? So SAR is basically synthesizing a series of compound, and then while changing few functional groups. So let's say in one compound I will change this group, in another analog I will change this group, and in another analog I will keep this script intact, and then I will change this one. So like this we synthesize a series of compound. For SAR's relationship studies we have to synthesize at least 30 to 40 compounds, and then by looking at the structure, by changing minor determination, we actually try to understand which groups are very important for the biological activity of that particular compound, and which groups are not that much important, means which groups we cannot change and which groups we can change. That is about the structure activity relationship studies. For example, in this structure you can see there are 3 methoxy groups here. So this methoxy group, it is said to be important for the binding ability. If I change this methoxy group with another group which cannot bind, or let's say I just keep this position only at hydrogen, so what we will see? We will see there will be a decrease of the drug's activity. This methoxy group is not much required, and this methoxy group is important for the correct conformation and activity. Here this methoxy group, it permits the halide, alkyl, alkoxy. So in this position, halides, alkyl, alkoxy, amine, SME, these are allowed but not bulky groups. Okay, so here this double bond is allows the ring contraction, this position, so it is important. And here you can see there is the epimer. Okay, this epimer diminishes in the activity, so we cannot have a epimer here, and this group can be replaced by other substituents. So as you can see, there is a normal bond here. If there will be an unsaturation, that unsaturation will increase the activity, and the ring expansion is not permitted here. So that's how you can see that the structural features of that particular compound, it is determining whether the compound will show a particular biological activity or not. Okay, so that is all about the structure activity relationship. So we have another example of SAR. So this is a compound, okay, which is a RET inhibitor. RET is a kinase. Okay, so as you can see, there are various structural features. So in this group here, polar groups are more preferred here in this position, this purplish position. N-methyl pyrazole and methyl sulfonyl phenyl groups are the best for the inhibition. Okay, so here the sulfur in the place of this nitrogen diminishes the activity severalfold, and methionation of the nitrogen also diminishes the activity. In this blue portion, 5 tertiary butyl butyl isoxazole and 3 tertiary butyl N-methyl 1H pyrazole are the most favored in this position. And in this red position, 1,3 substitution pattern completely attenuates the activity. So as you can see, these black positions, these groups are not changed. So whenever we are doing the structural activity relationship, so what we will do, we will synthesize a series of compound keeping some structural features intact, and let's say this core, this heterocyclic core and few features, this we will keep them intact, and we will change the functional groups which are in the periphery of that compound basically, and then we will check if the activity, the structural changes are favored by the biological activity shown by compound or not, and that is the structure SAR study. So what is SAR study? So SAR study basically the study of the structural features of a drug that are important to its biological activity. Okay, so number C was the correct option for problem number 1. So now I will move toward the next problem. So problem number 2 says SAR shows that important binding groups in morpholine are phenol, aromatic ring and amine. A diagram can be drawn which shows these functional groups and their relative orientation. What is the term for this diagram? Number A, pharmacodynamics. Number B, pharmacokinetics. Number C, chromophore, and number D, pharmacophore. So, what is the term for the diagram which shows the functional groups and their relative orientations that is important for the binding of a particular drug? Your options are pharmacodynamics, so A, number B, pharmacokinetics, number C, chromophore, and number D, pharmacophore. Again, I will give you some time. Read the question very carefully, read the options carefully, and then let me know which of the following is the correct term. SAR shows the important binding groups in morpholine are phenol, aromatic ring, and amine. A diagram can be drawn which shows these functional groups and the relative orientation. What is the term for this diagram? Pharmacodynamics, pharmacokinetics, chromophore, or pharmacophore? Let me check the chat box now. Okay, so there are answers about this question, number D, and number A. So we have different answer again. So let me check which of the following is the correct one, and then we can discuss it. So the correct answer of this question will be number D, pharmacophore. So pharmacophore is the term which is used to determine, which is the name of the diagram that can be drawn showing functional group and their relative orientation. Now let us understand each and every term which are mentioned in the options. So first is the pharmacokinetics and pharmacodynamics. So okay, the first is the let's say it is the pharmacokinetics. Let us discuss it. So pharmacokinetics is the quantitative study of the drug movement in and throughout the body. It is a study of processes by which a drug is absorbed, distributed, metabolized, and eliminated in the body. The absorption, distribution, metabolism, excretion of a drug involve all passages through cell membranes. Pharmacokinetic properties are affected by the route of administration and the dose of the administered drug. So pharmacokinetics, we have discussed a lot about pharmacokinetics in last week, about the bioavailability, about the pharmacokinetics pathway, about absorption, distribution, and metabolism and excretion, that is the ADME, which are the pharmacokinetics. And pharmacokinetics is basically what our body is doing to a particular drug. So a drug is going to get absorbed in our body, then get distributed, and then getting metabolized and excreted. So that is the study which is known as the pharmacokinetics. Then we have pharmacodynamics. Pharmacodynamics is the study of the mechanism of action of drugs, and that explains what does to the body and how they do it. It tries to explain the action, effect, and relation as well as the dose relation effect. It involves the drug receptor interaction, to elicit the action of the drug in the body. Pharmacodynamics is the study of the biochemical and physiological effect of a drugs and their mechanism of action, and it provides the basis for the rationality of use of drugs and design of effect for better drugs. Okay, so pharmacokinetics is basically the study explaining what the particular drug is doing to our body. Okay, that the effect of the drug on our body, what is the drug effect relationship, and it involves also the receptor and drug interaction, if there is a particular receptor or any protein or any enzyme, if the drug is acting as an inhibitor or a agonist or antagonist. All of these studies are done in case of the pharmacodynamic studies. Then we have the chromophore. So basically chromophore is a Greek word which is consists of chroma, which means color, and phoros means bearer. So it is defined as any isolated covalently bonded group that shows a characteristic absorption of the electromagnetic radiation in the UV or visible region. Compound containing chromophore are known as chromogen, like C double bond C, C double bond O, nitro group, etc. And then we have pharmacophore. So pharmacophore is an ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interaction with a specific biological target and to trigger or block the biological response. In drug design, the term pharmacophore refers to a set of features that is common to a series of active molecules. Hydrogen bond donors, acceptor, positively negatively charged group, and hydrophobic regions are typical features, and these features are known as pharmacophoric groups. So pharmacophore is the basically our correct, the right option, which is the pharmacophore, which is the ensemble of steric and electronic features to ensure the optimal supramolecular interaction or binding interaction to trigger the biological response. Okay. So very good, many of you have given the correct answer, which is good. So now I will move toward the next problem. So problem number 3 says, which of the following major aims in the drug design is not related to the pharmacodynamics of the drug? Your options are: number A, reduction of the side effects; number B, maximization of activity; number C, reduction of the toxicity; and number D, maximization of the oral bioavailability. So which of the following major aims in drug design is not related to the pharmacodynamics of the drug? Which of the following major aims in the drug design is not related to the pharmacodynamics of the drug? Number A, reduction of the side effect; number B, maximization of the activity; number C, reduction of the toxicity; and number D, maximization of oral bioavailability. Again, let me know in the chat box which of the following you think is the correct one: A, B, C or D. All of you please participate, even if your answer is wrong, it does not matter, but all of you must try to answer this question. Let me check the chat box now and then let us discuss about it. Okay, so I have multiple answers for the question. So your options are like number D, number D, number D, number D. Okay, so all of you think it is the number D which is the correct answer for this question. So now let me check if it is the correct one or not. So the correct answer will be yes, number D. The maximization of oral bioavailability, it is not related to the pharmacodynamics of the drug. So basically what is into the pharmacokinetics and pharmacodynamics? So basically the drug administration, the drug absorption, drug distribution, followed by the drug metabolism to the site of the action and the drug elimination, all are these into the part of the pharmacokinetics. Okay? And then from the transfer of to the site of the action to the drug, to the regulatory proteins, to receptor, enzyme, ion channel transporter, then binds to the molecular target, the affinity, the mechanism of drug action and response to the efficacy. All of these are the part of the pharmacodynamics. So what is that? Does it mean that as I have already mentioned, the pharmacokinetics is what the drug is our body is doing to the drug. Okay, so and then followed by what the drug is doing to our body. So the maximization of oral bioavailability, it is not a part of a pharmacodynamics, it is a part of the pharmacokinetics. If you remember the last in the last class where we were discussing about the bioavailability, I have mentioned that there are several ways of administrating a drug for the maximization of bioavailability. The drug can be given intravenously, in the oral manner, in the sublingual and nasal, etc. So there are various ways by which a drug can be administered for the maximum bioavailability, but the thing is this is part of the absorption or the administration, or which is into the pharmacokinetics, but not the pharmacodynamics. But the reduction of the side effect, the maximization of activity and reduction of toxicity, all these are aimed for the pharmacodynamics of the studies of the drug. So what are the aims and objectives? So pharmacodynamics can be defined as the study of the pharmacological effect of the drugs and their mechanism of action. The objectives of the analysis of the drug action are to understand the interactions between the drug and the target cells and to characterize the full sequence and scope of action of each drug. Okay. So question of problem number D, answer number D was the correct answer, which all of you have given correctly. So very good. So now I will move toward the next problem. Problem number 4 says cocaine was isolated as the active principle from coca leaves. Studies into SARs showed that the important functional groups for binding are those shown in blue, this one. So what binding interaction are likely for the aromatic ring? Your options are number A, hydrogen bonding as hydrogen donor, number B, hydrogen bonding as hydrogen acceptor, number C, van der Waals interaction, and number D, ionic bonding. So which kind of bonding interaction are likely to be shown by this aromatic ring? Again I will repeat, cocaine was isolated as the active principle from coca leaves. The studies into the SARs shows that the important functional groups for binding are those shown in blue. What binding interaction are likely for the aromatic ring? And your options are number A, hydrogen bonding as hydrogen donor, number B, hydrogen bonding as hydrogen acceptor, number C, van der Waals interaction, and number D, ionic bonding. Again take some time, read the question carefully and then read the answers carefully and let me know in the chat box A, B, C or D which of the following is the correct option. Okay, let me check the chat box now. Okay, so there are many answers are given, I think number C, number C, number C. So I can see that not all of you have given the answers till now. So I will give you 2 more minutes. All of you please try to answer the question, okay. Even if your answer is wrong, it doesn't matter, but answering the question is very important, otherwise I won't understand if you are following the class or not. So again I will repeat the problem number 4. Cocaine was isolated as active principle from coca leaf. Studies into the SARs show that the important functional groups for binding are those shown in blue. What is the binding interaction which is likely for the aromatic ring? Number A, hydrogen bonding as hydrogen donor. Number B, hydrogen bonding as hydrogen acceptor. Number C, van der Waals interaction, and number D, ionic bonding. All of you please try to answer the question. I will wait for 2 more minutes and then I will show you the answer and we will discuss it. Again I am saying, if you have any doubt or any query at any point of the session, please let me know in the chat box. For smooth streaming of the session, I have muted you all and kept your cameras off because I am recording the session. So so that there is no any network disruption, I have done it, but the chat box is always open and I am checking the chat box, okay. So if you have any doubt, any query at any point of the session, if you want to repeat me any question or anything, just you can let me know in the chat box. I will do it surely at the end of the session. So please try to answer the questions, and if you have any problem, let me know in the chat box. Okay, now let me check the chat box again and then I will discuss it. Okay, so I have few more answers. So it is said to be number C. Now let me check if the number C is the correct one or not. Okay, so the correct answer is very good, number C, van der Waals interaction. So basically, so whenever there is a aromatic ring, the intermolecular bonding can involving the aromatic ring can be the van der Waals interaction, or it can be induced dipole interaction, okay. So there are few interaction by which the aromatic ring is involved. So van der Waals interaction is one of those. So there can be also pi stacking interaction and the induced dipole interaction. So all of these are part of a van der Waals interaction itself, okay. So what is in case of the pi stacking? So basically the pi stacking which are the intermolecular interaction, the energy is from 0 to 10 kilocalorie per mole, okay. And this is a weak electrostatic interaction between the aromatic rings. There are two general types, one is face to face and one is edge to edge, okay. The face to face pi stacking responsible for the slippery feel of the graphite, and the similar pi stacking interaction help to stabilize the DNA double helix. So as you can see, if two aromatic rings are like this, the face to face, the pi electrons of one aromatic ring is interacting with the pi electrons of the other, okay. And this is known as the face to face pi stacking, and there is a face to edge. In case of here, the pi electron cloud of one particular aromatic ring is interacting with the aromatic hydrogen, okay. So this is also known as the phase 2 your H2 phase, the pi interaction, okay. So apart from pi stacking, we also have a pi cation or induced dipole interaction. So what does it happen? So basically the pi, there is a whenever there is a aromatic ring like this phase to phase, so they have the pi electron clouds. So all these pi electron clouds are the negative in charge, okay. So but if there is any positive cation like the ammonium ion or any kind of another cation, that particular ion, positively charged ion, can interact with the pi electron cloud which is negatively charged, which is known as the induced dipole interaction. So which is another kind of intermolecular interaction involving the aromatic rings. So option number C was the correct one. Van der Waals interaction is likely for the interaction for the aromatic ring in the cocaine, okay. So now I will go to the next problem. So problem number 5 says combristatin is an anti-cancer agent discovered from an African plant. Analog 1 is more active, than combristatin, whereas analog 2 has less active. Which strategy is used to designing the analogs 1 and 2? Again I will repeat, combristatin is an anti-cancer agent discovered from an African plant. The structure is this, okay, the combristatin. The analog 1 is more active than combristatin. This analog 1 is more active than combristatin, whereas the analog 2 is the less active, okay. So which strategy is used here in designing the analog 1 and 2? Your options are: number A, simplification; number B, isosteric replacement; number C, rigidification; and number D, chain contraction. So which of the following is the correct strategy which is used in designing the analog 1 and analog 2 for the combristatin? Simplification, isosteric replacement, rigidification and chain contraction. Again, look at the structures very carefully, read the question very carefully, and then try to answer the question in the chat box. I will wait for some time for 2 to 3 minutes, and then I will show you what is the correct answer after checking the chat box, and we will discuss about it. So this combristatin is a very important natural product, and it is a very potent anti-cancer agent which is discovered from the African plant, okay. And there are several analogues of the combristatin itself. And this combristatin is also, I have discussed it earlier also, I think. So it is a tubulin polymerization inhibitor. Take some time, take 2 to 3 minutes, read the question and let me know A, B, C or D, which of the following is the correct answer. Let me check the chat box now, then I will discuss this answer. Okay, so there are several answers. Number C. So number C is the most given answer. Let me check now if the number C is the correct one. So the correct answer will be yes, it is the number C, rigidification. So the rigidification is basically, so as you can see this bond, it is a single bond here, right. But when there is a elimination of water from this position, there is a hydrogen, right. So if there is a elimination of water, so what we will have? We will have this double bond here. So this is called the rigidification strategy. Why it is called the rigidification? Because this bond is single bond and this bond is free to rotate, right. So because this bond is free to rotate, it can acquire any kind of conformation, different conformation. But when we just have a double bond here, right, like this then we are just fixing it, right. It can be either Z isomer, which is this one, or E isomer, which is this one. So this Z isomer, it is known as the combristatin A4, which is the more potent anti-cancer agent than combristatin itself, but the analog 2, which is the E isomer, is not that much potent as the analog A, because of some sort of this particular orientation of the groups. Okay. So the technique which actually the strategy which restricts the rotation of the particular compound by fixing the particular conformation and configuration of the drugs, it is known as the rigidification technique. Okay. So now next problem. Problem number 6 says succinylproline was the lead compound for the captopril, which acts as an inhibitor of an angiotensin converting enzyme. What is the relevance of the thiol group? Number A, it can form a covalent disulfide bond with the cysteine thiol groups in the active site. Number B, it acts as an isostar of the carboxylic acid. Number C, it acts as a bioisostar of the carboxylic acid. And number D, it is a smaller group than carboxylic acid and fits to the binding site easily. Again I will repeat it, succinylproline was the lead compound for the captopril, okay, which acts as an inhibitor of ACE enzyme. The ACE enzyme inhibitor means it is a potent antihypertensive drug, which we have discussed in the last class itself. So what is the relevance of the thiol group which is present in the succinylproline? Now the options are: number A, it can form a covalent disulfide bond with cysteine thiol group in the active site. Number B, it acts as an isostar of carboxylic acid. Number C, it acts as a bioisostar of carboxylic acid. And number D, it is a smaller group than the carboxylic acid and fits in the binding site very easily. Read the question carefully, read the options carefully, and then let me know in the chat box A, B, C or D which of the following is the correct answer. Let me check the chat box. Okay, I think I have few answers. So number A is the most given answer. Now let me check if number A is the correct one or not. So the correct answer is number C. It acts as a bioisostar of the carboxylic acid. Okay. So basically, so this kind of bioisostar. So what is a bioisostar? So let me tell you first about the bioisostar. So bioisostars are the substituents or groups with similar physical or chemical properties which produce broadly similar biological properties in a chemical compound. In drug design, the purpose of exchanging one bioisostar to another is to enhance the desired biological or physical properties of the compound without making the significant changes in the chemical structure. Okay. So, for example of the bioisostar, we will I will show you this one also again. So, this is aminopyridine, which is a marketed analgesic and anti-inflammatory drug in 1896, and this aminopyridine was known to be carcinogenic later in 1922. So, what is so then we have this propylphenazone, which is the bioisosteric modification of this aminopyridine. Well, what it had done, that it has this bioisosteric modification of this dimethyl amino group is removed, because this group was responsible for the particular carcinogenic action. Okay. So, similarly, in case of the succinyl proline also, the lead compound for the captable, the relevance of the thiol group is like this: thiol group can act as a bioisostar of the carboxylic acid. Okay, because free carboxylic acid groups are not very much tolerable in case of the particular drug. I will show you later also what is the different bioisostar of carboxylic acid. We will discuss it about later, but just remember, free carboxylic acid groups are not very much good tolerable in case of the drugs in the lead compounds. Okay, because the free carboxylic acid group, they participate into several several interactions which are not favorable for the ADME property, also for the metabolic stability of a drug. Okay. And there is also differences between the isostar or bioisostar, which I will discuss later in today's class itself. So, now I will go to the next problem. Problem number 7 says, what is the reason for the improved activity of the B rather than A? So, this is the structure of the B. Okay. So, it is said to be that B has an improved bioactivity than A. Okay. So, look at the structures carefully. So, what is the difference? The difference is there is no nitrogen is present here, but there is a nitrogen present in the B. Okay. So, then which is marked here also. So, what is the reason of the improved activity of B rather than A? Your options are: number A, less toxic metabolite formation; number B, increased solubility; number C, increased bioavailability; and number D, good pharmacophoric profile. What is the reason of improved activity of B rather than A? The difference of A and B, which is marked here again. So, there is a nitrogen group is present here in the B, which is not present in the A, and the nitrogen group maybe it is somehow reason for the improved activity of the B rather than A. So, A, B, C or D, which of the following seems to be the most logical reason for the improved activity of B rather than A? Number A, less toxic metabolite formation; number B, increased solubility; number C, increased bioavailability; and number D, good pharmacophoric profile. Think and then let me know in the chat box: A, B, C or D, which of the following is the correct answer. me check the chat box and then let me discuss about it. Okay, number D, number B, number D, number A. So, I have different answers for this question. Okay, let me check which of the following is the correct one and then let me discuss about it. So, the correct answer will be number A, less toxic metabolite formation. So, now let me show you. So, basically it is a HIV-1 attachment inhibitor, okay, means which is a anti-HIV drug. So, as you can see, this is the structure A, right. This is the analog A and this is the analog B. So, when we are changing this particular ring to this ring, means whenever we are adding a nitrogen here, so the aqueous solubility of the group is increasing and the toxicity is decreasing, and means it is a better improved activity, and this is a BMS 488043 which is in a phase 3 of clinical trial. So, what is the reason? So, basically the reason is whenever this structure A, this drug is metabolized in vivo means inside the cell. So, what happens, because of this OMe group and OE group, these are hydrolyzed to the hydroxyl groups, and these form the toxicophore, which is a quinone analog. Okay. But whenever there is a nitrogen here, if the quinone is formed, so this is a quinone, this is a non-toxic amide which is a metabolized form. Okay. So, this quinone is toxic, but this amide is non-toxic. So, since we are changing this particular ring to this, adding up this nitrogen, so adding up this nitrogen, it removes the toxicophore, which is the quinone, from the system. That is why the toxicity is decreasing and the aqueous solubility of the drug is increasing, and the activity of the drug is the improved activity of number B rather than number A because of this less toxic metabolite formation. Okay. So, remember, whenever there is this kind of group, they are prone to form, very much prone to form the toxicophore, which is the quinone one. Okay. So, because of this, this kind of structure are not favored much in case of the drug designing system. Okay. So, now I will go to the next problem. Problem number 8 says, which of the following is the most frequent protein ligand interaction? Your options are number A, hydrogen bonding, number B, pi-pi interaction, number C, ionic interaction, and number D, hydrophobic interaction. Which of the following is the most frequent protein ligand interaction? Hydrogen bonding, pi-pi interaction, ionic interaction, or hydrophobic interaction? Which of the following is the most frequent protein ligand interaction? Hydrogen bonding, pi-pi interaction, ionic interaction, or hydrophobic interaction? All students, please try to answer the questions. Okay, otherwise I won't understand if you are interacting or if you are participating in the class or not. So, A, B, C, or D, which of the following is the correct one? Again I am saying, if you have any doubt, any query, please let me know. Okay, let me check the chat box and then let me show you what is the correct answer. Okay. Number A, number A, number D, number A. Okay, number A is the most given answer, followed by a number D. Okay, so let me check which one of the following is the correct one, and then let me show you the correct answer. So, the most frequent protein ligand interaction is hydrophobic interaction. So, let me show you the particular frequency distribution curve for the most common non-covalent interaction observed in the protein ligand. So, basically the interactions are, these are the non-covalent interaction. Okay, non-covalent means there is no bond formation, but there is a covalent interactions. Okay, so non-covalent interactions are like, the most frequent one is the hydrophobic interaction, followed by the hydrogen bonding interaction. Then we have the pi-pi stacking interaction. Then we have weak hydrogen bonding interaction, followed by the salt bridge interaction, amide stacking, and then cation pi interaction. Okay. So, the most frequent interaction is the hydrophobic interaction. Okay. Because in case of the protein ligand interaction, what happens basically, so there are various structure of the protein. So, basically it is the, there are various group which are interacting with the particular ligand in the hydrophobic manner. Okay. So, hydrophobic interaction is the most important interaction. So, remember this. Most of the student I have seen that they are thinking it is a hydrogen bonding interaction. I have seen it this year also, in last year also, that hydrogen bonding is the most given answer, but the correct answer will be hydrophobic interaction. And this is the almost twice the large, as the interaction, the frequency is twice as large as the for in case of, in comparing to the hydrogen bonding one. Okay. I will show you the example of hydrophobic interaction in the next slide itself, or in only, I think. So, the correct answer was number D, hydrophobic interaction. So, now I will move toward the next problem. So, problem number 9 says, by which method chimeric drugs are formed? Your options are: number A, in silico drug designing method; number B, pharmacophoric hybridization method; number C, bioisosteric replacement; and number D, structure based drug design. So, by which method chimeric drugs are formed? In silico drug designing method, pharmacophoric hybridization method, bioisosteric replacement method, or structure based drug design method. Again, all of you try to answer the question in the chat box. A, B, C or D, which of the following you think is the correct one? First you try, then I will show you the answer and we will discuss about it. Now let me show you the correct answer and then let us discuss about the method for having the chimeric drugs. First let me check the chat box. Okay, so the answers are given like number B, number C, number A and number B. So again I have multiple answers. So now let me check which of the following is the correct one. Okay, so the correct answer will be number B, pharmacophoric hybridization. So it is the method by which the chimeric drugs are formed. So what are the chimeric drugs? So basically our method to synthesize or design a chimeric drug that contains key pharmacophoric features of a true drug. So basically chimeric drugs are a mixture which have the pharmacophoric features or the important structural features of two different drugs. For example, a structure containing features of true methoxy, estradiol and colchicine has been synthesized as a potential anti-cancer agents. Although both are the parent structure have anti-cancer activity, they have serious drawbacks like 2-methoxyestradiol is metabolized rapidly while colchicine has a toxic side effect, but the chimeric structure also has anti-cancer activity, but it has improved pharmacokinetic properties. So this is the 2-methoxyestradiol and this is the colchicine. So both have different structural features. So this what does this chimeric drug has done in this pharmacophoric hybridization method? The important pharmacophores or the important structural features are incorporated in a single drug to form a particular chimeric drug which has the similar or improved anti-cancer activity along with improved pharmacokinetic profile. Because the parent drug they have serious drawbacks like the 2-methoxyestradiol is metabolized rapidly and colchicine has a toxic side effect, but this chimeric drug have a improved pharmacokinetic property, and since the key structural features or the key pharmacophoric feature of two different drugs are incorporated in a single drug, the method is known as the pharmacophoric hybridization. Okay. So number B was the correct answer. So now moving to the next problem. So problem number 10 says DU12290 is more potent and selective than saltopride because, so this is the DU12290 and this is the saltopride. Okay. So this compound is more potent than this one because number A, pyrrole ring has made it more rigid. Number B, replacing the amide moiety has improved the lipophilicity. Number C, both A and B are correct. Number D, it cannot be said for sure only from the structures. This is the saltopride, this is the DU12290. This DU12290 has improved activity, means it is more potent and selective than the saltopride. And what is the reason? Your options are number A, pyrrole ring has made it more rigid. Number B, replacing amide moiety has improved the lipophilicity. Number C, both A and B are correct. And number D, it cannot be said sure for sure only from the structures. So A, B, C or D, which of the following is the correct one? Let me know in the chat box. Look at the structures carefully, read the options carefully and then let me know which of the following is you think is the correct one or the correct answer for this problem number 10. Let me check the chat box now. Okay, number A, number C, number C. So I have 4 number C's and 1 number A. So now let me check which of the following is the correct one, and then we can discuss it out. So the correct answer will be number A. Pyrrole ring has make it more rigid. So that is the reason why Du-122-290 is more potent and selective than saltopride. The pyrrole ring of Du-122-290 serves to increase the rigidity of the side chain present in the saltopride. The red colored bond, so there is not I have not mentioned it here, so just let me show you. Yeah, this red colored bond which is here in saltopride, this one actually, so this is freely rotable, okay, but it is locked when there is a pyrrole ring. In case of pyrrole ring, this bond is locked, but here in case of saltopride, it is freely rotable, right. This reduces the number of possible conformations of Du-122-290 can adopt, and it increases the chances of the active conformation being present when it enters to the binding site, thus it increasing the activity. Moreover, some of the conformations that are now disallowed may be those which are adopted when the saltopride binds to the D2 receptor, thus increasing the selectivity for the D3 receptor. So basically saltopride is mainly for the binding for the D3 receptor, okay, but when there is a less selectivity for the D3 receptor, because since saltopride has a active they can adopt many active conformations, so some conformations were able to bind to the D2 receptor itself, but in case of Du-122-290 compound, since this group is not freely, this bond is not freely rotable, it has fewer conformation which increases the selectivity toward the D3 receptor rather than the D2 receptor, okay, and this makes this compound more potent than saltopride itself. So problem option number A was the correct one. So now next problem. So problem number 11 says procedure of transforming racemic drugs to single enantiomer is called number A chiral pool, number B asymmetric synthesis, number C chiral switch, and number D asymmetric switch. So what is the procedure of transforming racemic drug to single enantiomer is called? Chiral pool, asymmetric synthesis, chiral switch, or asymmetric switch? Procedure for transforming racemic drugs to single enantiomer is called? Which will be the correct answer? Procedure of transforming racemic drugs to single enantiomer is called? I will give you 2 more minutes and then I will show you the answer after checking the chat box, and we will discuss it. Now let me check the chat box. Okay, the answers are given like number B, number B, number C, number D, number C. Okay, so again I have multiple answers for the question. Now let me check which of the following is the correct one, and then let us discuss about it. So the correct answer will be number C, chiral switch. So basically, what is the first, let us understand what are the advantages of the chiral drug. So chiral drug is a single agent instead of a mixture of two distinct drugs. Simplifies the interpretation of the basic pharmacology, greater selectivity for their biological targets, improved therapeutic indices and reduced adverse effect, longer or shorter duration of action, more appropriate and dosing frequency. Okay, so chiral switch is the procedure used to transform an old racemic drug into its single active enantiomer. The new enantiomeric drug developed by the pharmaceutical manufacturer will receive the additional patent protection and a new generic name. Okay, so the chiral switch is the procedure, remember this, that is used to transform an old racemic drug to its single active enantiomer. So now next problem. Problem number 12 says tetrazole is a bioisostar of which group? Number A, phenyl group, number B, carboxylic acid group, number C, ketone group and number D, amide group. So tetrazole, which is a heterocyclic nitrogen containing heterocyclic ring, we all know it has 4 nitrogen in a single in a 5-member ring. Okay, so it is a bioisostar of a particular group, and which is a very important bioisostar of a group. So which is the group for which it is a bioisostar? Number A, phenyl group, number B, carboxylic acid group, number C, ketone group and number D, amide group. A, B, C or D, which of the following is the correct one? Think and then let me know in the chat box. Tetrazole is a bioisostar of which group? Phenyl, carboxylic acid, ketone or amide? Let me check the chat box now. Okay, I have answers. Number D, number B, number C, number B, number C, number B. Okay, number B, number C is the most given answer, I think. I have number D, I think. Okay, let me now check which of the following is the correct answer and then let us discuss about it. So, the correct answer will be number B, carboxylic acid group. So, isostars of carboxylic acid have been studied extensively, and these studies have typically focused on enhancing potency, reducing polarity, increasing lipophilicity to improve the membrane permeability, enhancing the pharmacokinetic property and reducing the potential for toxicity. As I have mentioned earlier also, carboxylic acid group is not very much good for the activity or the pharmacokinetic profile of a particular drug, okay, because the drug will be not much lipophilic, and the lipophilicity is important for the improving the membrane permeability of a particular drug. That's why the isostars of carboxylic acid have been studied extensively, okay, and there are various bioisostar of the carboxylic acids, okay. For example, there are common isostars or these groups, okay, and the heterocyclic based isostars are these. In this the groups which are highlighted here are the most important bioisostars of the carboxylic acid, and one of this is tetrazole, which is a heterocyclic based bioisostar. So, there is an example, okay. So, the example is angiotensin 2 receptor antagonist provide the instructive insight into carboxylic acid bioisostar design. Since binding affinity to a receptor in a series of biphenyl acids are sensitive to identify the acidic element, okay. So, this compound this is the lead compound and this is the R. So, during the SAR studies, okay, for the angiotensin 2 receptor antagonist is a hypertensive agent, the activity, this is the IC50 value. So, if there is a carboxylic acid group, the IC50 is in nanomolar is 275, okay, but if there is this kind of group, okay, so the as you can see the IC50 value is decreasing, and the decreased IC50 value, the more the IC50 value is lesser, the more the compound is more potent. So, as you can see, increasing the changing the carboxylic acid group to tetrazole have decreased the IC50 value from 275 to 3 nanomolar, okay, which is a huge difference. So, which gives an insight why carboxylic acids group are not very much good for the biological activity for a drug, and why tetrazole is a very important bioisostar of the carboxylic acid group, okay. So, now moving forward, I will go to the next question. So, problem number 13 says why B is more potent inhibitor than A. So, this is the B which has the inhibition constant of 1 nanomolar, but A has inhibition constant of 39 micromolar, which is a huge difference is there, you can see. So, why this particular B is more potent than A? Okay, options are number A, hydrophobic interaction is more, number B, hydrogen bonding interaction is more, number C, van der Waals interaction is more, number D, all of 3 options are correct. Why B is more potent inhibitor than A? Hydrophobic interaction is more, hydrogen bonding interaction is more, van der Waals interaction is more, or all of these options are correct. A, B, C or D, which of the following will be the correct one answer? Or the correct reason for B to be more potent inhibitor than A? Let me check the chat box now. Okay. Number A and number D. We have most the answer given is number A, and then we have a number D also. So, let me check which of the following is the correct one. So, the correct answer will be number A, hydrophobic interaction is more. So, as you can see, we have a hydrophobic group which is present here in B, and this hydrophobic group is reason for which the hydrophobic interaction is more in case of B, otherwise the structure are same. Because of this hydrophobic group, the hydrophobic interaction will be more, and as we have seen earlier that the frequency of hydrophobic interaction in case of the drug receptor interaction is the highest one, since then because of that increasing hydrophobic interaction will increase the potency of the inhibitor. So, this is the protein structure of the, this is the ribbon structure of the protein, and this is the group. So, this is the protein ligand interaction diagram. Okay, we will discuss about this kind of diagram in our summary session, which I will take in the last. Okay, this is not directly involved in your course, but these things are very important for the medicinal chemistry. So, which I will discuss at the end of our course, which is in the summary sessions. Okay. So, now let me go to the next problem. Problem number 14 says, which of the following statements is true? Options are: number A, the aromatic rings can interact with quaternary ammonium salt through the dipole induced dipole interaction. Number B, the aromatic rings can interact with the quaternary ammonium salts through the induced dipole interaction. Number C, replacing aromatic ring with cyclohexane will have no impact on the interactions. And number D, the interaction is also called a pi cation interaction. So, which of the following statement is true? Number A, the aromatic rings can interact with quaternary ammonium salts through the dipole induced dipole interaction. Number B, aromatic rings can interact with quaternary ammonium salts through the induced dipole interaction. Number C, replacing aromatic ring with cyclohexane will have no impact on these interactions. And number D, this interaction is also called pi cation interaction. Which of the following statements is true? Read the options carefully and then let me know which of the following answers are correct one. Let me check the chat box now. Okay, so I have several answers: number A, number B, number A, number C, number A. So, number A, B and C are the most given answer. Okay, now let me show you the correct answer for problem number 14. So, the correct answer will be both number A and number D are the correct one. Okay, now let me show you. So, basically first let us understand what is a dipole induced dipole forces. So, basically it is between a polar and non-polar molecule. This type of forces operate between a polar molecule which has a permanent dipole and a non-polar molecule whose electron density is symmetrical. A polar molecule may sometimes polarize a non-polar molecule, thereby deforming its electronic cloud which lies in its vicinity. So, in this manner it induces a dipolarity in the non-polar molecule. Okay, so this is a permanent dipole which is a polar molecule, and this is a non-polar molecule. Sometimes this permanent dipole of a polar molecule can induce a dipole of a non-polar molecule, which is known as the dipole induced dipole forces. The interaction energy is inversely proportional to the 6th power of distance between two molecules. Okay, and the induced dipole moment depends on mainly on two factors. One is dipole moment, which is present in the permanent dipole or molecule, which is the polar molecule, and polarizability of the electrically neutral molecule, which is the non-polar molecule. Molecules of larger size get polarized easily, thereby the strength of the attractive interaction increases, and dispersion forces also operate in such cases. So, cumulative effect of dispersion force and dipole induced dipole interaction exists. That means the total effect is greater than the sum of the individual effects of the dispersion forces and dipole induced dipole interaction. Okay. So, for example, there is a benzene ring or pi cloud and there is a potassium cation, okay, and this is known as the pi cation interaction. Okay. So, the induce another possibility, so this particularly whenever there is a ammonium ion, okay, and there is a this particular your pi electron cloud. So, and one possibility is induced dipole interaction between the quaternary ammonium ion and any aromatic ring in the binding site. The positively charged nitrogen can distort the pi electrons of the aromatic ring such that the dipole is induced, whereby the face of the ring is slightly negative and the edges are slightly positive. Okay, and then this allows the interaction between the slightly negative faces of the aromatic ring and positive charges of quaternary ammonium ion. This is known as the also known as the pi cation interaction. Okay, so induced dipole interaction is also known as a pi cation interaction, where there is a positively charged cation is induced to the dipole or distorts the pi electron clouds of an aromatic ring. Okay. So, both option number A and D were correct in this case. So, now next problem number 15 says which of the following statements is false regarding the lactams. Your options are: number A, they act as an acylating agent and irreversibly inhibits the bacterial enzyme by acylating a serine residue in the active site. Number B, they can form intermolecular hydrogen bonding. Number C, lactam can undergo chemical reaction with the target if the ring size is large and no strain is suffered. And number D, a prominent example of lactam ring is present in the penicillin. Which of the following statement is false regarding the lactams? A, B, C or D? Again I will repeat. Number A, they act as a acylating agent and irreversibly inhibits the bacterial enzyme by acylating the serine residue in the active site. Number B, they can form intermolecular hydrogen bonding. Number C, the lactam ring undergo chemical reaction with the target if the ring size is large and no strain is suffered. And number D, a prominent example of lactam ring is present in penicillin. So, A, B, C or D, which of the following statement is false regarding the lactams? me check the chat box now. Okay. Number A, number C, number A, number C, number C. So, number C is the most given answer, followed by number A. Let me check which of the following is the correct one. So, the false statement will be number C. The lactam ring can undergo chemical reaction with the target if the ring size is large and no strain is suffered. This is the false regarding the lactams. So, basically this is called the alpha lactam, this is called the beta lactam, this is called the gamma lactam, and this is the delta lactam. Okay, and which is because of the number of carbons are present. So, if there is a one alpha carbon is alpha lactam, then we have one alpha and one beta, then we have alpha beta gamma, and then we have alpha beta gamma and delta. Okay. So, amides which are within the ring system are called the lactams. They too can form intermolecular hydrogen bond. However, if the ring size is small and suffers ring strain, the lactam can undergo chemical reaction with the target, leading to formation of a covalent bond. Best example of these are penicillin, which acts as an acylating agent and irreversibly inhibits the bacterial enzyme by acylating the serine residue in the active site. So, all penicillins contain 4-member beta lactam ring. The ring contains 2 carbon atoms and sp3 hybridized and nitrogen atom which is sp3 hybridized and a carbon which is sp2 hybridized. The structure is similar to the combination of 2 amino acids cysteine and valine. Okay. Because of the restriction of the ring, normal bond angle of 109.5 and 120 are not able to be obtained. So, the ring is strained. This makes amide highly reactive. The ring opens to the penicillin can become covalently bonded to the enzyme that synthesizes the cell walls of bacterium, thus blocking its action. Okay. So, option number C was the correct one, which is mostly given by you all. So, now next problem. Problem number 16 says which of the following is not an isostar of methyl group. Your options are number A hydroxyl OH, number B NH2, number C SH and number D oxygen. Which of the following is not an isostar of CH3 group? Number A hydroxyl, number B NH2, number C SH and number D O. Let me check the chat box now. Okay. So, number D, number A, number C. Again I have multiple answers. Let me check which of the following is the correct one. So, the correct answer will be number D, oxygen. Oxygen is not an isostar of methyl group. So, isostars are atoms or group of atoms which share the same valency and which have chemical and physical similarities. For example, SH, NH2, CH3 are isostar of OH, whereas sulfur, NH and CH2 are isostar of O. Isostars can be used to determine whether a particular group is an important binding group or not by alternating the character of the molecule in as controlled way as possible. And examples of bioisostars is like aminopyridine and propylphenazone, which I have mentioned earlier also. Okay. So, there is a difference between isoelectronic and isostar. Okay. So, the term isoelectronic refers to having same number of electrons or the same electronic configuration. By isostar is the term isostar refers to the chemical species having same similar size, the same number of atoms and the same number of valence electron. Chemical species, atoms, ion and molecules. Okay. And ions and molecules can be the isostar. Similarities, electronic configuration for the isoelectronic one, and that isostar molecular ionic size and have a similar number of atom and valence electron. For example, carbon monoxide gas and nitrogen gas are isoelectronic to each other, and benzene and thiophene are isostaric to each other. Okay. So, problem number D was the correct answer. Number D was the correct answer for the problem number 16. So, now moving forward, problem number 17, it says that in which of the following ways 3D pharmacophores of flexible structures can be achieved. Number A, rigid analogues of the flexible compound could be synthesized and tested to see if wire activity is retained. Number B, crystallize the target with the compound bound to the binding site. Number C, NMR spectroscopy to solve the active conformation of isotopically labeled molecules bound to their active site, and number D, all of the above. In which of the following ways 3D pharmacophores of a flexible structures can be achieved? In which of the following ways 3D pharmacophores of a flexible structures can be achieved? Number A, rigid analogues of the flexible compound could be synthesized and tested to see where the activity is retained. Number B, crystallize the target with the compound bound to the binding site. Number C, NMR spectroscopy to solve the active conformation of isotopically labeled molecules bound to their active site, and number D, all of the above. Now, let me check the chat box and then I will show you the correct answer. Okay. So, the answers are given like number A, number C. So, number A is the most given answer, followed by the number C. So, now let me just show you the answer and let us discuss about it. So, the correct answer will be number D, all of the above. So, basically a pharmacophore is a specific 3D arrangement of functional groups within a molecule framework that are necessary to bind to a macromolecule and or an enzyme active site. The identification of pharmacophore is an important step in understanding interaction between the receptor and a ligand. 3D pharmacophore is a 3D dimensional pharmacophore. It specifies a special relationship between the groups, and it is expressed as the distance angle, distance range, angles and planes. A commonly used 3D pharmacophore for antihistamine contains 2 aromatic ring and a tertiary nitrogen. So, identifying 3D pharmacophores is not a very difficult for rigid cyclic structure. For example, glipin exam, so we looked at when you flexible structures, then the problem is not as but when you have a flexible structure, the problem is not as straightforward. At what happens, we all know there is going to be a large number of shapes and conformation for a molecule to likely take up. So, then become more complicated to interpret, because important binding groups may be actually particular conformation which are active conformation. If they are not present in the conformation, then the molecule may not active. So, again we can look at this in bit in more detail later, but one may safely assume that only one or may few conformation of a molecule is recognized and bound to the binding site. So, if you have a linear molecule, it becomes little more bit complicated to make the interpretation of this concept. So, the number of ways in which this can be achieved is rigid analogues of the flexible compound could be synthesized and tested to see whether they are activity retained. Crystallize the target with the compound bound to the binding site, and NMR spectroscopy to solve the active conformation of isotopically labeled molecule bound to their binding site. So, these are the ways by which a 3D pharmacophores can be achieved for a flexible compound. Okay. So, now next problem number 18 says, which of the following characteristic is necessary for an ideal lead compound? Your options are: number A, poor activity; number B, significant side effects; number C, good selectivity; number D, difficult synthetic protocols. So, which of the following characteristic is necessary for an ideal lead compound? A, B, C or D? Which of the following characteristic is necessary for an ideal lead compound? I think this is a very easy question and all of you should answer it correctly. Let me check the chat box. Okay. The number C is the only given answer. So, I think all of we know that it is the correct one. So, yeah, number C, good selectivity is the necessary characteristic of an ideal lead compound. So, discovery of a lead compound is one which has a number of attractive characteristics such as desirable biological activity, but may have other undesirable characteristics such as high toxicity, insolubility, metabolic problems. The ideal lead compound is modified by alternating the structure to get desired activity and to eliminate the side effects. So, very few compounds are ideal, most likely to have low activity, poor selectivity and significant side effects. They may also be difficult to synthesize. So, there is an advantage of finding analogues which are improved properties. Strategies that can be used to optimize interaction of the drug with its target in order to gain better activity and selectivity are therefore important. So, strategies that can be used to optimize interaction of a drug with target helps us to gain better activity, and perhaps the process we may also get better selectivity, and therefore, it is very important to exercise that we can use in drug design. Okay. So, now question number 19, The structure number 2, which is this one, was found to be more interactive with target rather than the parent compound number 1. Okay, what is the reason? Number A, the strategy used was one of the ring expansion. Number B, blue nitrogen allows better solubility. Number C, blue nitrogen can act as a hydrogen bond donor. And number D, the strategy used could be described as the extension, since there is a binding interaction has one further binding interaction has occurred. So, which is the correct reason for this compound 2 to be more active than this lead compound 1? Number A, the strategy is used is one of the ring expansion. Number B, blue nitrogen allows better solubility. Number C, blue nitrogen can act as a hydrogen bond donor. And number D, the strategy used could be described as the extension, since the further binding interaction has occurred. Let me check the chat box. So, answers are given like number C and number D is the most given answer. Now, let me check which of the following is the correct one, number C or number D, or there is any other correct answer. So, number D is the correct answer. The strategy used could be described as an extension, since a further binding interaction has occurred. So, one advantage of altering an aromatic ring to heteroaromatic ring is that it introduces the possibility of an extra hydrogen bonding interaction with the binding site, and it should be a suitable binding region available. This is the extension strategy. For example, structure 1 was the lead compound for a project looking for novel antiviral target. Replacing aromatic ring with a pyridine ring resulted in an additional binding interaction with the target enzyme. That is why this number 2 is the more potent and more interactive than number A. Okay. So, answer number D was the correct answer for the problem number 19. So, now last question of today's session. It says introducing a methyl substituent to dopamine antagonist results in a dramatic reduction of affinity. What is the role of methyl group here? Number A, metabolic blocker. Number B, configurational blocker. Number C, conformational blocker. And number D, beta blocker. Introducing a methyl substituent to a dopamine antagonist results in a dramatic reduction in affinity. What is the role of methyl group? Metabolic blocker, configurational blocker, conformational blocker, or beta blocker? Let me check the chat box, since we do not have much time left for today's class. Okay. So, the answers are given number C, number A, number D. Okay. So, again I have multiple answers, number A, number C and number D also. So, let me check which of the following is the correct one. So, the correct answer is number C, conformational blocker. Okay. So, what is a conformational blocker? Another tactic that has the same effect as used is the use of conformational blocker. In certain situation, a quite simple substituent can hinder the free rotation of a single bond. Introducing a methyl substituent to the dopamine D3 antagonist gives structure 2, which results in a dramatic reduction in the activity. So, as you can see, there is a structure number 1 and then there is a structure number 2. Now, if I in this bond is for free rotation is easily this biphenyl system, but if I introduce a methyl group here, this free rotation is hindered. Okay, so in structure 1, free rotation around the connecting bond allows the molecule to adopt a conformation where the aromatic ring are coplanar and the active conformation for the receptor. In this case, the conformational blocker rejects the active conformation. In other cases, an increase in activity is also possible. A steric clash between new methyl group and orthoproton of the neighboring ring, which prevents both rings being the same plane. Free rotation around the bond between two rings is no longer possible, and so the structure adopts a conformation where two rings are at a particular angle to each other. Okay, so it is known as the conformational blocker. So, this was the last question of today's session. So, what we did in today's class is we discussed many things about the optimizing the the topic was the optimizing the drug target interaction. So, we discussed many topics regarding those. We discussed about bioisostars, we discussed few about the SAR and the pharmacophoric interaction. So, this was the week 7 live session and we discussed about the week 8 course content. So, now I will be sharing my email ID with you here in the chat box. Okay, so if you have any doubt, any query, okay, one second. So, yeah, in the chat box I have shared my email ID. So, if you have any doubt, any query, feel free to contact. Okay, let me know what is your doubt or what is your query. I will try to answer this those queries. So, today it is it was a week 7 live session. So, this kind of live sessions we will be having on each Tuesday from 6 p.m. to 8 p.m. Please try to join on time and attend the sessions. Okay, it will be helpful for your solving the assignment and understanding the course better. So, now I will be closing the session for today. Thank you all for joining and let us meet on the next session. Okay, and I will share this class PPT and the video soon within the and you can check it from the SWAYAM portal. Okay, after some time. So, I will close the session for today. Thank you all for joining. Thank you all. I will just close the session for today.